East Coast. So we'll get started. So, hello. Thank you for joining us today for our monthly Tick IPM Working Group webinar. Today we are lucky to have Dr. Kyndall Braumuller from University of South Carolina to present on her work titled Engorged Data, a snapshot of South Carolina's tick surveillance program. And just a reminder of who we are, the IPM Institute was founded in 1998 with the goal to increase adoption of IPM in agriculture and communities using educational and solutions-based approach. The organization is split into two groups: the Sustainable Food Group, which focuses on food supply chain sustainability, in many cases works directly with growers, and the group that I work in, the Sustainable Communities Group, which focuses on sustainability education and resource development as it relates to people, pets, and pollinators in community spaces like parks, fields, schoolyards, and buildings. How we make a difference? We have a number of programs, including a Green Shield Certified certification, which is a program with the mission to reduce the use of pesticides and provide best use practices for pest management professionals, facilities, and landscape companies. The Pest Defense for Healthy Schools, which focuses on educating K through 12 staff about IPM and best practices, limit exposure and impact of students, with free courses on our website. And the Tick IPM Working Group, which, as you know, focuses on integrated tick management and education around ticks and tick-borne diseases, hosts annual virtual Tick Academy, and produce educational resources, among other things that we do. And as always, this work is supported by the North Central IPM Center. And here is just our funding acknowledgement. And a quick update on the Tick Academy is that it is less than 2 months away. So our early bird pricing ends on Sunday, August 31st. So register now. It also informs us if you want CEUs, so we could better provide CEUs for you and whoever else is interested in participating in Tick Academy. We have our 2 days of October 15th and 16th. 16th, we're still finalizing speakers, but we have the focus on outreach and management the first day, and then medical and veterinary focus on the second day. And now to introduce our speaker today, Dr. Kyndall Braumuller is a research assistant professor in the Department of Epidemiology and Biostatistics within the Arnold School of Public Health at the University of South Carolina. She earned her Ph.D. in epidemiology at the University of South Carolina, where her research focused on tick-borne spotted fever group rickettsioses in El Salvador. Dr. Braumuller has a wide variety of vector-borne disease epidemiology, ecology, and outbreak investigation experience, including in tick-borne spotted fever group rickettsioses, mosquito-borne Crosse Encephalitis virus, West Nile virus, malaria, and triatomine-borne Chagas disease. Her current research builds on her over 10 years of experience specializing in the field, where she strives to understand more about vector-borne disease dynamics in South Carolina and abroad to ultimately lead to advances in preparedness and prevention. So without further ado, I'm going to stop sharing, let Dr. Braumuller share, and I will, in the transition, I will put up the pre-webinar question.
Okay, can you see the slide? I'm gonna make sure that everyone can hear me and you can see my slide. Okay, awesome. Okay, well, thank you so much for the invitation to give this talk. I'm very excited to kind of share what we've done here in South Carolina, and I've just been a small part of that. But thank you again for inviting me, and I'm happy to kind of share this with everybody. So like Zach said, I do a lot of medical and veterinary entomology, mostly medical, but and I did do a lot of tick stuff for South Carolina. I am dabbling a lot more in mosquitoes lately, but I am happy to share also some of my colleagues' work here from South Carolina. Let's see. Okay. So I'm gonna kind of describe just the snapshot of what we're doing here in South Carolina, and I have to start from the beginning, from scratch, basically what we started to do. I'll go through some of our ticks that we've collected, some of our studies, and I'm going to really highlight a rickettsia study specifically, and also alpha-gal that we've started to do some work with as well. Briefly on some tick-borne virus information, and kind of very quickly talk about the CDC Center of Excellence and kind of our current collaborations. So typically South Carolina is thought about actually in a lot of ways with chronic disease, cardiovascular disease, obesity. We have pretty high rates of opioid-related deaths or outbreak related to cardiovascular disease and these things. We have a lot of STIs in South Carolina. In short, we have a lot going on, but one avenue that has historically not been investigated or been glossed over is vector-borne disease. And our neighboring states, specifically like Tennessee and North Carolina, in particular, had a lot more publications, a lot more information about ticks and tick-borne disease prior to 2020 when I started in 2019 here. And so I'm going to kind of go back in time to talk about this prior to 2020 period. I was tasked with starting the tick surveillance project basically for the state, and I was able to help do that. But we had to really start from scratch. There was no local or state investment in tick surveillance at the time. There was very little published information on tick species or pathogen prevalence or presence. And that's where we kind of started. So we had to think at that time, where were vector-borne diseases reported or where tick-borne diseases were reported from the health department. In 2019, at the time, approximately 70% of our vector-borne diseases were tick-borne reported by the health department. And counties with the largest urban centers had our highest cases of Lyme disease that were reported. And the circles are showing those urban centers. On top, you have the Greenville area. Center is Columbia. On the right of the state is Myrtle Beach, and on the south is Charleston. But we did see counties with more rural populations had higher rates of spotted fever group rickettsioses that were reported by the health department at the time as well. So what we did was we wanted to try and hit rural and urban kind of centers to try and see if we could cover as much of the state as possible. Our state health department separates the state into 4 health regions. And on the left map, those are our health regions. We have the upstate, Midlands, on the right is PD, and then on the bottom is low country. So in 2019, 2020, we just went out and we selected state parks, as many as we could try and think we could surveil in a year, with just me and another person, basically. So those are the green triangles are our state park locations where we attempted to collect ticks. The red squares are veterinarian locations or animal shelters that we reached out and we had actually people say that they would collect ticks for us. So we started off pretty small, but we wanted to hit all 4 health regions with multiple parks and multiple animal shelters. We really had no resources in the beginning. Dr. Melissa Nolan, who is my PI, she devoted discretionary funds for this. We both used our personal vehicles to drive around the entire state for this. She used her discretionary funds for purchasing surveillance tools and kits, the first rounds of pathogen testing, things like that. We also utilized collaborations pretty heavily to try and get this up and going. And lastly, we had a lot of undergraduate volunteers and a few graduate students who are our surveillance team leaders. You can see we're wearing face masks in that middle picture because, of course, 2020 was when COVID happened and we were still trying to surveil for ticks in the parks. And at the time, you know, there wasn't really much. We weren't sure what the regulations were going to be. So we were asking everyone to wear masks, even though we were outside if they were going with other people. And so it really was a challenge to recruit students for this, but we were able to get students more and more every single year and kind of work towards establishing this program, especially when we partnered with our state health department and other partners, like I said, and I'll mention them. We don't have an entomology program at the University of South Carolina either, so in order to get students excited about field work, sometimes we have to promise them lab work as well. And so we're trying to, balance between doing field work and lab work and things like that. But we know that the field is where the ticks are, so we need to get out there and get them first. So, in 2020, we only surveilled for ticks from March through October, but then after 2021, we now do 12-month surveillance. And we have done some changes for what our tick surveillance protocols are. But in general, we tried to follow the CDC recommendations for tick dragging. We also included tick traps, which are our carbon dioxide baited traps, which is that picture right there. And then our animal shelter submissions. At first, we didn't really have a way to do it other than I was literally driving to animal shelters and handing them mason jars filled with ethanol and then getting like the top right picture. And then we eventually created these kits that we would send out through the mail that had ethanol instructions, parafilm, things like that we had, and then a log, of course, for our volunteers to write down animals that they were collecting ticks from. And we actually got some state vehicles at one point, I think in 2022, so we were able to stop using our personal vehicles as well. And in 2020, our reach, so let me move my sharing screen so I can see my notes. In 2020, we were able to surveil about 12 counties, and now in 2025, we're able to reach across the entire state. So we only have 46 counties, but we're able to actually surveil the entire state, which has been much better. We've also added even more since this was published. This is a picture from a publication that I'll include the title from in a couple slides. But this kind of gives a better idea of the coverage across the state that we now have. And this map does not include the various locations from which we've had public submissions as well. So in addition to us going out, we also have people send us ticks, and which has helped us cover the entire state. But you can see each little paw print is a shelter or a veterinarian office that collects ticks for us. And then the little trees are parks that we now surveil. So our range is much wider across the state. And that's because we've had so much help from students and our partners. And so the ticks that we've actually collected, we've gotten, I think, 8 different species. And I think we got 8 different species only our second year that collected, and that hasn't changed yet. So our second year, we actually collected the first Haemaphysalis longicornis ticks that were collected in the state. And so that's why I bolded that one. We collect about a couple thousand ticks a year, and Amblyomma americanum heavily takes up maybe 95% of the ticks that we're collecting. And very quickly about Haemaphysalis longicornis. In 2020, we found 1 adult female in both Pickens and Lancaster counties, which border North Carolina. And so those were our first reports of this tick in South Carolina. But in June 2022, we had 13 of these ticks, nymphs, submitted to our lab for identification from 1 location in York County. And this was the first established population of Haemaphysalis longicornis in the state. So this was actually a cattle farm in York County, like I mentioned, which does border North Carolina. It's in between Lancaster and Pickens, which is a little bit further west. And this image is an image of that cattle farmer's farm that we visited. And this is just one of his fields. And it's interesting that this one field is where the ticks were. We went to multiple other fields of his, and we did not find these Asian longhorn ticks. It was literally only in this very localized area. So what we did is we conducted drags around the perimeter of this one field, and we did 6 defined plots within the field. We conducted blood meal analyses on the ticks, and then we tested, we live collected them and froze them so that we could test them for anaplasma, phagocytophilum, Borrelia burgdorferi, and Thaleria orientalis. So we got almost 2,000 of these Asian longhorn ticks in less than 4 hours from this one field, which was pretty staggering. For the blood meal testing, we saw that about 36% of the pools that we tested, because we tested them in, they were almost all nymphs, so in pools of 5 nymphs, 36% of them had fed on dogs only. 30% fed on cattle and 15% fed on people. And then there were some additional like dog and cattle or dog and human feeding as well. We also saw that ticks from the perimeter had larger diversity of blood meal hosts, and this is not surprising. We did get 2 positive pools for Thaleria orientalis and 5 pools positive for Anaplasma. And we did not sequence the pathogen, so we don't know if this was the Ikeda strain of Thaleria orientalis, though. We continue to monitor the situation as it evolves. We're interested in working with USDA and our partners, specifically Clemson, our agriculture hub, for additional funding for control of these ticks. We're interested in keeping on working with them because we know that this situation is going to continue to get worse, and we are expecting the ticks to continue to spread across, especially that northern part of the state. And so now I'll briefly talk about some of our rickettsia studies. So the initiation of this statewide program really opened up a lot of doors for our group, and we've been able to explore epidemiology and biology of ticks and tick-borne diseases in the state. I don't have time to go into all of these, but I wanted to touch on one specific example from my colleague, Dr. Lydia Gual-Gonzalez, who did her dissertation on spotted fever group rickettsioses in South Carolina. So I'm going to touch on one of her papers, left paper, exploring the eco-epidemiology of Rickettsia amblyommatis and Rickettsia parkeri in ticks from South Carolina. So, but we've been able to look at some human data, and that's what that top paper looks at. And then also another one of our students looked at incomplete feeding behaviors, where we did additional blood meal analyses on specific ticks. But overall, we've been able to collect so many ticks for the past 5 years or so that we have quite a bit of data, and we're continuously thinking of new ways that we can look into this information. I'm hoping to do some phylogenetic analyses with our partners at University of Tennessee and continuing with our pathogen studies and communicating with other individuals in Tennessee. So going into Lydia's paper, and I did steal these slides with her permission. So specifically for spotted fever group rickettsiosis, we know that rickettsii is our most pathogenic, which is the causative agent for fever. And Rickettsia parkeri is somewhat new, and its primary vector is Amblyomma maculatum. But we also have Rickettsia amblyommatis, which falls within this debated pathogenicity group. Multiple people have said it does cause disease in humans. It certainly infects people. But does it cause disease? Yes and no. And so it is pretty heavily distributed across our Amblyomma americanum population. We see a lot of this pathogen in a lot of our Lone Star ticks. Human and animal studies have described infections through antibody tests or IFAs, but it's pretty difficult to determine which pathogenic agent is causing infection. We have cross-reactivity with a lot of these assays, these antibody tests. And so we were interested in looking at some epidemiological studies to test ticks using molecular detection. And we have found that Rickettsia rickettsii is pretty rare in our tick populations. And so where are all these spotted fever group rickettsiosis cases? Where are they in the state, and what is actually causing them? If we're not seeing Rickettsia rickettsii, is it parkeri, or is it potentially amblyommatis? And so Lydia and us, and we were interested in understanding the different species interaction, its effect on disease epidemiology, and seeing if we could look at all of these together in our ticks that we've collected across the state. So we wanted to understand the geographic spread of the different Rickettsia species and elucidate regions where exposures could happen. And we wanted to evaluate whether different species interact and how the environment might be playing a role in their distribution. So for this study specifically, we used data from 2021 to 2022 in South Carolina. We had ticks in this study specifically from those years from South Carolina state parks and animal shelters. And so we collected the questing ticks using the density dragging and carbon dioxide traps, which I mentioned earlier, and also host-attached ticks were submitted from animal shelters or veterinarians. These ticks were identified to species, life stage, and sex, and then pooled by date and location. And the way that we pooled was up to 3 adults in one pool, 5 nymphs in one pool, and all larvae from one location basically were pooled together. And host-attached ticks were pooled as singular ticks, so not pooled. We estimated rickettsia prevalence and performed some Bayesian regression models to estimate infection based on environmental factors and a correlation analysis evaluating rickettsia co-presence between the pools. So the Bayesian analysis, it used environmental variables defined by land use classification, raster data from the National Land Cover Database, and we extracted information on the land classification around 10 kilometers buffer within the state parks, and we imputed the quantitative values in a Bayesian logistic regression model in R. So for some of our primary findings, for this study in particular, there were almost 3,000 ticks questing and from animal shelters. 99.5% of those were Amblyomma americanum, which is not a surprise, and 25% had Amblyomatis, and 19% had about our Parkeri. Sorry, I'm describing the questing ticks only. And then for the host-attached ticks, we had 52% Dermacentor variabilis and about 30% Amblyomma americanum and 13% Amblyomma maculatum. And you can see the distribution of the Amblyomatis and Parkeri in those ticks as well, which is a little bit different, but much higher Amblyomatis in that group. So most questing ticks came from the southern coastal plains in especially Edisto State Park. You can see that very large red circle. Edisto State Park, if any of you have been to South Carolina, it's a beautiful park. It's on the coast, and it is where we get a lot of ticks. And so we also get a lot of ticks that are infected with pathogens, multiple different types of pathogens in Edisto State Park or along that coastal region. So we saw host-attached ticks were higher from the Piedmont region, and so that would be sort of towards the north end of the state, those larger green circles. So we get quite a lot more ticks from our animal shelters or veterinarians from the upstate region of the state versus that coastal region. So interestingly, we found some antagonistic associations or what we would call negative correlations between Amblyomatis and Parkeri positivity. We found Amblyomatis was more prevalent in our Lone Star ticks and in the dog ticks and R. sanguineus as compared with Parkeri, whereas Parkeri was more present among our Amblyomma maculatum ticks, which that was not surprising. But other correlations were found for life stage and location. This may have been described previously in controlled conditions in laboratories where rickettsia acquisition was lower in rickettsia infected ticks, and it's harder for those infected ticks to actually maintain 2 species of pathogen through transovarial transmission. And so some of these studies have found that during co-feeding, acquisition of Parkeri is less likely if the tick is already infected with Amblyomatis. Finally, what we saw with the environmental variables was that several factors influenced one species over the other differently. Amblyomatis presence was positively impacted by tick sex, collection method, and negatively affected by presence of emergent herbaceous wetlands, while Parkeri presence was not affected by the same variables, but was positively affected by herbaceous vegetation, cultivated crops, and woody wetlands. So it's been described that increased suitable habitat presence correlates with greater species diversity, and thus enzootic cycles have more opportunities to propagate. Based on our findings, we could think that the forests could potentially act as pathogen influencers, while pasture and crop fields sort of reduce maybe transmission risk, which would be due to a lesser species diversity and potential use of acaricides in those monocultures. In the region where we most collect, we have emergent herbaceous wetlands, which are more suitable for amphibians and reptiles, and we potentially have lower pathogen presence. And that maybe we hypothesize that these animals are poor propagators of these pathogens, and that's why we potentially have lower presence of amblyommatis in these same locations. So based on these results, since this was published, we have continued our surveillance studies, and we're hoping to potentially do this study again, but with more data, more longitudinal data, to examine this relationship further. The antagonism that we saw between amblyommatis and parkeri could have a regulatory role on pathogenic rickettsia species, and we know that this could be seen again with a larger length of data that we can look at. So we're excited to continue working on this and understanding how different rickettsia species interact across different tick populations. So we're looking forward to continuing that work. Okay, so I'm going to switch gears now to talk about alpha-gal, which we have in the past couple of years started to do a little bit of work with. And I also stole these slides with permission from Dr. Emily Owens-Pickle in our lab. She did her dissertation on alpha-gal here in South Carolina. So for those of you who are not familiar with alpha-gal, this is a sugar molecule or a carbohydrate that is in mammal species, except for simians. So monkeys, apes, and humans do not have this alpha-gal molecule inside of our muscle, basically. But it is found in all other mammals. And it's expressed in many parasites as well. So what's interesting about alpha-gal is that it is related to tick bites. And so what we've seen specifically in the United States is that the Lone Star tick in particular has this sugar molecule inside of its saliva. And when it bites someone and that saliva is injected into that person, and this person potentially has alpha-gal sensitivity, then they can develop what's called alpha-gal syndrome, which is an allergic reaction that their body mounts against the alpha-gal. And so not every person will develop this. This is an allergy, so it's heavily dependent on each person specifically in their immune system. But it's very, very interesting. So the symptoms of alpha-gal syndrome, specifically abdominal pain, nausea, diarrhea, vomiting, heartburn, indigestion, hives, and itching. But severe symptoms can occur like anaphylaxis and swelling of one or more of the lips, tongue, throat, etc., shortness of breath. So it can cause very serious impacts on people who do develop alpha-gal syndrome. And because it's an immune response, some individuals may be able to, if they stop consuming mammalian meat, potentially, they could eventually ingest it later in their life if they stop exposing themselves. However, some people have not been able to do that. And if you get continuously bitten by ticks, if you expose yourself, then you're potentially just increasing your body's reaction to this as well if you're already sensitized. So it's a very interesting dynamic that people are trying to learn a lot more about, especially here in the southeastern United States. So it's not just mammalian meat that has alpha-gal, but there are also some products that have been developed using mammalian origins. So antivenoms, some vaccines, some monoclonal antibody therapies for cancer, and some other things are actually produced with either mammalian origin information or something similar. And so people will still have this alpha-gal syndrome reaction if they are sensitized when they're not even eating mammalian meat. Like, for example, the cancer treatment drug. This was actually how we found out that this was linked to alpha-gal, because a lot of cancer patients were receiving this drug, and a very large proportion of those individuals started developing anaphylaxis, which was very strange. And so it kind of launched into this whole investigation. But if you're thinking, oh, well, I'm a vegetarian, I don't need to worry about getting alpha-gal, that's not necessarily true. A lot of products that are made with gelatin, so like gel caps in pills or something like that, can also trigger this response. So it is serious and very interesting, and we're hoping to learn more about it. And that's what Emily was able to do. So what she did as part of her dissertation was she used data as part of a large-scale study in South Carolina called the All in South Carolina study that was not related to alpha-gal, but was actually related to COVID-19, where we had participants who were recruited at public community events. They were from almost across the entire state of South Carolina. We administered a health survey and collected biological samples, primarily blood from these individuals, and we were able to test them for the alpha-gal sensitization. So serum testing was performed using an ImmunoCAP 250 system, which is a high throughput instrument widely used for allergen-specific IgE quantification. And participants completed a health survey, which included questions related to vector-borne disease exposure, demographics, and general health history. So this was her first hypothesis, and what she did was looking at specific demographics, environmental exposure factors. She did, these are the statistics that she listed, and I'm not going to actually show the data because she's still working on getting this written up for publication, but I can go through the very pertinent results from this. So what we saw were statistically significant risk factors related to alpha-gal sensitization in our South Carolina cohort. Was male gender was associated with alpha-gal, less than a high school degree or high school degree education. Less than a high school degree or high school degree education. Sometimes or often finding a tick biting themselves, which makes sense. And they found sort of this bimodal relationship with age, where the youngest age group and also the oldest age group had a statistically significant association with alpha-gal sensitization. So what was surprisingly not significant was individuals who said that they spent time outdoors that was not associated with alpha-gal. There were no differences among race, and there was no differences among employment status or like part-time versus full-time, when we were expecting to see something related to outdoor time especially. And we commonly see a lot of infectious diseases and especially chronic diseases associated with race in our state, but we didn't see that with alpha-gal. And so for our hotspot analysis, she also looked at where alpha-gal sensitization was particularly most prevalent. And we had about 4 counties that were hotspots for alpha-gal in the state. So we saw some regional clustering, the southwestern part of the state, and we didn't have any cold spots of significance, only 1 hotspot. And so for those not familiar with South Carolina, this part of the state is very rural. We're not close to really any large urban centers. I feel like the closest, maybe Savannah actually, or Beaufort, South Carolina, which is where Hilton Head Island is. So primarily we're seeing a large number of individuals with the highest amounts of sensitization in rural southwestern part of the state, which is very interesting. And this is just another way to kind of view that same data of the density of sensitization across the state. And you can kind of see where we had a lot of people participating for this study. But you can see that dispersion across the state is relatively even. We're not reaching those far corners, but that same density within that same corner we're still seeing. And we're not seeing a relationship with urban centers at all. So this is related to our rural areas. Columbia, Charleston, and Greenville, if you remember where those are, they're not heavy hitters in this alpha-gal density at all, which we find very interesting. Okay. And again, I'm going to take another step to kind of talk about some tick-borne viruses. This is also preliminary data that Emily is working on publishing. So this hasn't been officially finalized yet, but we're pretty excited about what this is looking like. Hopefully you are familiar with Heartland and Bourbon virus. They're not related to alpha-gal really, but they are driven by the same vector, which is the Lone Star tick in the United States. And they're important novel tick-borne viruses with quite a bit of, or with a very low amount of research because there's not been very many cases. So Heartland was detected from 2 cases in 2009 in Missouri. That's when we initially found out about it. But it can cause pretty serious disease if one is infected. And we saw antibody prevalence high in white-tailed deer and raccoons. In South Carolina, so in animals, there's been antibodies to this virus. And it's potentially transstadially and transovarially transmitted in our Lone Star ticks. And a national study saw across 7 states, there's a pretty high hospitalization rate. 2 out of 14 have died from infection, but many people do get an acute infection, acute disease from this as well. And for bourbon virus, it's relatively newer in terms of how long we've known about it, but from the same region of the United States, Missouri, Kansas, patients have presented with a similar symptomology. But this is the first thogotovirus detected in the Western Hemisphere, potentially also transmitted transstadially and transovarially as well. What we did with this study was she used also ticks that we've just collected over the past couple of years with our surveillance project that this is established. These videos are of Emily herself doing some of the testing. So we've identified all these ticks and stored them, and she was able to do these additional virus tests on them with ticks that were collected live. So we did start live collecting ticks I think in 2022. So previous to that, I didn't mention it, but we would only put ticks in ethanol, but now we collect ticks live and keep them on a cold chain so that we can test them for a virus. So we pooled our ticks by 5 from site data collection and collection method. We had positive controls for both viruses provided by the CDC Arboviral Reference Collection, and we used RT-PCR in order to test for both of these viruses using protocols that have already been described. So we did find positive results for Heartland virus, but no bourbon virus. And so again, these haven't been officially published yet, but she is working on this. So I'm not going to go too far into the details. But across 2 years, we did see some interesting pool results. And we had some pools that coincided with a peak of Lone Star and nymphal activity. So some of these ticks were infected from drags or from CO2 traps. So we've seen them collected from different methods. And we don't know if this is a significant finding or not, but 4 out of 6 of our Heartland positive pools were also correlated or co-infected with Rickettsia parkeri. So we don't know if that's well, it is interesting. We don't know if it's important, but we thought that was fun. And so we're continuing to look into this, and we are going to continue testing our samples for these viruses as well. Again, no positives for bourbon, but we potentially did find Heartland in our ticks. And where we saw these ticks was, again, in that kind of coastal region, very close to Edisto State Park that I mentioned, but also the other park in Beaufort County is, I'm forgetting the name of it, but just that same coastal region. So you can see it's the same like southern coastal plain where we're seeing a lot of activity down there. So we're going to continue looking at this low country region of the state because we think that there's something going on with pathogen interaction, pathogen prevalence, and also the number of ticks that we can collect down there is pretty high. And so we're interested in continuing to really look at this coastal region of the state. So currently, we partner with the CDC Southeastern Center of Excellence in vector-borne diseases. We have great relationships with our rickettsial diseases branch. We work with Old Dominion University, the Tennessee Department of Public Health, Georgia Department of Public Health, Florida International University, and the University of Florida. And I'm only naming institutions that I know we've done tick work with. We have other partnerships as well at the Southeastern Center of Excellence. Other partners that we work with are our State Department of Public Health. We work with the Clemson University, especially our state veterinarian and entomologists. We worked with University of Tennessee, Knoxville, UNC Chapel Hill, and NC State University, all related to tick work. And we couldn't have done any of these things without our partners. So we're very grateful for the collaborative nature and the helpfulness of everyone that we've worked with. And we hope to continue working with each and every one of these individuals and groups. Right now, like I said, we do year-round tick surveillance and pathogen testing. We have over 20 people now who help do this work. A lot of undergrads still, but now we've got lots of masters, PhDs, and kind of everyone helping, which has been really wonderful to kind of take this program to the next level. We're continuing our pathogen surveillance. We are doing quite a bit of sequencing as well now, and we're hoping to do some next-gen sequencing like to examine pathogen movement potentially within the southeastern United States. We're now also interested in looking at some microbiome studies and genomics of the tick species themselves within the southeastern US. So because we're collecting just this wide breadth of information, and we've been able to for the past 5 years. We're really excited about the things that we can do with it. We also, like I mentioned earlier, accept tick submissions from the public, and we welcome those from across the state for identification and pathogen testing. We've had a lot of great relationships with other people as well when people send us cool specimens, and we can kind of educate the public as well. We're also doing some tick attractant studies, part of the CDC Center of Excellence, continuing to monitor the Asian longhorn tick presence in the upstate. And yeah, so that's currently our program. We started at a very small number of people using personal vehicles, discretionary funds, and where we could go basically. And we didn't let COVID stop us from, going all over the state. Now we partner with a lot of great institutions and a lot of great people. So I hope that was informative and you kind of got to see a little, like I said, a little snapshot of what we're doing here in South Carolina. And if you're a part of our partners watching, thank you guys so much. And it was great to talk about this. So thanks.
Thank you so much, Dr. Braumuller. Real quick before we get, or as we transition to Q&A, I'm going to put up the post-webinar question. And it looks like one of our co-hosts, Nikki, had a question that she put in the chat. Um, is it more likely that people with a high school education or less being more likely to develop alpha-gal syndrome? Is that due to their work outside in rural areas? Although I think you said that it's not associated with just being outdoors more. Can you add a little more clarification?
Yeah, absolutely. We didn't go further than that question, so I don't know the actual answer to that. So because the parent study was a COVID study and we were able to kind of sneak in some tick-related questions, we were able to kind of tease at that, but we don't actually know. So that's a really great point. So yeah, we did see the lower education as associated, but working outside was not. So I don't know the answer to that, but that's a really good question.
Oh,
Awesome.
thanks. That'll be interesting to look into more.
Yeah, I definitely agree. We have a great relationship with our rural health center in the Arnold School, and I've kind of floated that question, like maybe we can reach out more with our rural health partners to try and tease at that a little bit more.
Awesome. So now to start going through the Q&As, Laura Rothfeld asked I had quite a few questions that seemed like you answered, but one of them was if she could reach out to you about starting a program in Arkansas.
Yeah, absolutely. My email is on one of these slides, and I think it was probably Oh, I was looking at it. Sorry. That's my email. Please reach out. Yeah.
And to answer your question in the chat as well, Laura, the goal is to have this presentation posted on the Tick IPM Working Group's YouTube page within a month or so, but give or take some time given some personnel changes. Another question from Leah Colton. This is, I think, in reference to your first portion of your presentation. If the collected ticks were nymphs, did they feed on the host as larvae, or were they not nymphs but mixed populations?
Um, is that for the Hemophysalis longicornis, like the blood meal testing? Is that what that question is referencing? Sorry.
Yes.
Okay. So it's probably leftover blood meal remnants from the larval stage, if I am thinking of it correctly, because they were questing when we collected them. So it was most likely from the larval stage.
Okay. Got it. Got it. Let me just clear those. And then a question from Zoe. How many Kieranzi do you see relative to SCAPs, if you have a
Oh,
chance?
yeah, that's a good question. So we definitely get a lot more scapularis than current. I don't know how to. I always say it wrong. Currency. We get actually more currency in the southern portion of the state, and we kind of see scapularis all over. But I would say we probably see well. Yeah, of the Ixodes, Kieran probably is about 15 to 20%. The scapularis is much higher.
Awesome. Another question from the Q&A portion, and I think you should be able to see it now. From Jenny Hall, in your study of Asian longhorn ticks, I believe York County, you mentioned 50% only fed on humans. Can you expand on that? How are you able to determine that? And do you have more supporting evidence of this tick biting humans?
No, that's a good question. I have to think back to this study. So when we did it, when we used the PCR, oh, I have to go back to the paper. Sorry, I probably can't answer it completely. Um, But yes, we had only found a relatively small number that had only fed on humans. So we only found evidence of the gene that was specific to humans in those pools. Um, and I can't, if you go back to the paper, our methods are described, and I'm probably mumbling about this and not answering this eloquently. But it was dependent on which pools were positive for which genes that we targeted for each animal, including humans. And we used multiple different reference papers that did a very similar thing. So um, that was all we could say on whether or not the blood meal analysis came back for humans. So I know this tick isn't typically found on humans, but that is what we found. Sorry, I can't really answer that better.
Awesome. Um, yeah, thank you for the question. Um, I'll wait a few more minutes if there's any more questions. It, the only other update we have from my side, or the IPM group's side of things, is we just have the Tick Academy lined up in October, so please sign up for that. Um, and we'll wait a few more minutes for questions, but if there's no other questions, thank you, Dr. Braumuller, for presenting for us. Really appreciate the information you shared.
Yeah, thank you for inviting me again, and thank you to all our partners and everybody who helped.
Alrighty, I will close down the Zoom now. I'll see you guys soon. Thank you again. Bye now.
Thanks.